This meeting is being recorded.
Well, welcome everyone. We are glad to have you here. This is the Virginia Women in HPC Lightning Talk, Student Lightning Talk session. And we are excited today to have quite a few great talks, many of which I'm going to learn a lot because I'm not at all familiar with those areas. The way that we're planning on working this is each student will have 3 minutes to do their presentation. We would like them to briefly introduce themselves, quite briefly, because you don't have much time. Spend most of the body of your presentation talking a little bit about your work, what's the problem, what's your approach to the problem, and then end with, we'd like you to briefly talk about what's next for you. If you're nearing the end of your graduate career or undergraduate career, you can tell us if you're going to go to graduate school, if you're looking for a postdoc, or if you're ready to go out in industry. Tell us what you're looking for. And hopefully from that, we will be able to create connections to help you along the path of your career. So are there any questions? If not, our first speaker is Lakshmi Miller. Take it away, Lakshmi.
Thank you, Dr. Gardner. Is everyone able to see my slide?
Yes.
Hi, everyone. I'm Lakshmi Madhavan Miller. I work for the VA-WHPC lab, and I perform research on autonomous underwater vehicles. AUVs, or autonomous underwater vehicles, are different from submarines in the sense that they're smaller in size and more unconventional in design and do not require a pressurized hull. Therefore, they're very versatile in applications. This also means that there are no people or nuclear power on the craft. And this implies that they do not have the restriction of the safe operating envelope that's required of submarines. Therefore, they can be used like a self-driving car. Their missions are very autonomous, but the problem is the same as self-driving cars, where I need to know where they're at all times. This brings me to the point that they need a very accurate maneuvering model. My dissertation concerns the CFD-informed lumped parameter maneuvering model of an autonomous underwater vehicle. CFD, or computational fluid dynamics, helps simulate the real-time physics of the craft underwater. This is done by solving Reynolds average Navier-Stokes equations, either in a time-steady or unsteady fashion. Such forces and moments obtained from those simulations are used to come up with hydrodynamic derivatives, which are then implemented in a lumped parameter model. Lumped parameter models are state space models, which means that they use these hydrodynamic derivatives and come up with positions, displacements, velocities, and orientations of the crafts at any given point of time. With that, I would say that I have made 4 clear contributions to existing literature in AUV maneuvering. 1, coming up with a novel way of producing hydrodynamic derivatives using RANs or URANs. 2, guidelines for simulating captive virtual planar motion mechanism maneuvers, which helps in coming up with the forces and moments that eventually become hydrodynamic derivatives. 3, definition of a new stability index in the vertical plane, which accounts for hydrostatic effects of the craft, which are usually neglected for larger and faster submarines. 4, hull-rudder interaction, which quantifies this quantity in a nonlinear fashion and implements this in the lumped parameter model. Throughout my time at VT, multiple crafts have been used to achieve these 4 things. But of these, the dive LD craft that you see on the screen everywhere is very special to me. The dive LD craft was designed by my advisor and I from scratch from a piece of paper with a diagram on it. The hull is 3D printed. It is uniquely shaped to be drag optimized at a specific speed, and it contains a drag optimized sail that's shaped like a NACA profile. Moreover, it contains 2 skates that you see on the diagrams on the left, and these facilitate parking the craft on land on a stand. Thus, my passion lies in coming up with abstract ideas and implementing them systematically. And this is observed by my projects, by my publication record, my professional service, and the background of the slide, which is like an engineering drawing paper. Thus, any job that lets me do this in a most efficient manner without a glass ceiling would be my ideal choice upon graduation. Right now, I have a tentative offer with the US Navy or the Naval Warfare Center to start as their controls engineer for submarine maneuvering. Thank you.
Thank you, Lakshmi. We have time for one question while the next presenter sets up their slides. Eric. Oh, you're clapping. I thought you had a question. Okay, Lakshmi, how is CFD in water different than CFD in air? I'm familiar with air, having worked in aerospace, but not at all familiar with water.
Okay, CFD in water is different in main two ways than air. One is it's incompressible. For the most part, I can avoid compressibility effects in at least deep water, not near the surface. And two, the density of water is different. Therefore, the turbulence modeling that's used for the speeds used underwater, which are much slower than the speeds used in air, would be different. I would be more concerned in using something like a transitional turbulence model, which helps me calculate the transition from laminar to turbulent flows, whereas in air, you need a basic amount of speed to take off. So you're not going to be in the regime of such slow speeds. So yes, the transitional model is also different.
Thank you. Thank you.
Thank you.
Really appreciate that, Lakshmi. Rashmi, are you ready?
Yes. Can you hear me?
We can. Please go ahead.
I'm gonna start. Can we start now?
Yes, please do.
Okay. Hello, everyone. My name is Rashmi Chawla. I'm second year PhD student in Aerospace and Ocean Engineering Department at Virginia Tech. I want to start my talk asking this question, that can we predict the future? We are not sure, but as engineers, I believe we should be able to predict the behavior of our engineer design. Even with tremendous development in computational mechanics, I believe we are still not there yet to fully eliminate expensive physical testing or guarantee safety relying only on our simulations. Over time, researchers have been developing robust computational techniques and addressing limitations. My research is also on one such technique called material point method, which is very well suited for large deformation. It is a meshless simulation technique. To demonstrate large deformation, I wanted to show this video, which was created for a Disney movie named Frozen, where MPM was used to simulate the behavior of snow. Qualitatively, we are using this technique to simulate the behavior of ceramics in thermal protection system in hypersonic devices. Some class of ceramics can withstand aerodynamic heating, which is of the order of 2000 degrees Celsius due to vehicles' very high velocity, which is approximately 5 times of normal passenger jet flying plane. Here is the demonstration of how we are incorporating thermal diffusion, oxidation at very high temperature, mechanical loading with continuum damage definitions. Moving forward, our approach is to develop multi-scale computational framework where we scale down from macro scale to meso scale to micro scale. At micro scale level, the effect of microporosity is taken into account and material properties are calibrated with the experimental results. Once these material properties are calibrated, we use those material property and verify at the mesoscale level. Currently, our 3D mesoscale code is under development. So this is one of our benchmark problem that we have created recently. Once the verification of material properties is complete at the mesoscale level, our next step will be to do the dynamic simulation for more realistic dynamic loading condition. This is one of the application of material point method. It has been under exploration for variety of materials, and I'm glad to be able to contribute to such a versatile technique and step towards accurately predicting the future of our creation. Thank you.
Thank you. So before we ask a first question, Rashmi, what is the next step for you after your degree?
So as I'm a second year PhD student, I would look for more collaboration for the research that has been going on in MPM. So because I have time to go out in the industry, this is my just second year. So I would look for more collaboration in this research field.
Thank you.
Yeah.
We have time for one question.
I would like to ask a question. This is Eric Walter. I was just curious. Since we're only doing like 2 or 3 minutes, maybe I missed this, but what is the range of target simulations you do? I saw you showed the Disney sort of the animation before, but what other what are the target areas where these simulations are most useful and give the best where are they typically deployed, these approaches?
Yeah, so this is actually a meshless technique, which is, I would say, take the advantage of finite element method. If you look for the research, other research is going on, it is this is very popular in geotechnical engineering, where people are using this to estimate the soil properties or find out the landslides. So for granular materials, it is doing well. For soft tissues, I have seen papers in it. So yeah, it is under exploration, and it became popular, I would say 25 years back, and it is been growing a lot in last 25 years.
Yeah.
Thank you.
Yeah.
Nina, would you please share your slide? I should have had you share as soon as Rashmi was done. Thank you.
Is everyone able to see my screen?
Yes.
Okay.
Go ahead, Nina.
All right. So, hello, everyone. I am Naina Pisharoti, a fourth-year PhD student at Virginia Tech, and the focus of my research is on high-fidelity computational analysis of UAV propellers. Now, let's break this title down. First of all, why UAVs? If you look at the 4 corners of the slide, you will see the various applications of UAVs. And mind you, this is not an exhaustive list. One of them being reconnaissance missions in the military, the next one being where companies like Amazon, UPS, or Pizza Hut, who are interested in doing drone deliveries. Aerial photography is also really picking up. And the final one, which is my favorite one, shows you a futuristic view of what our urban areas would look like once we have flying taxis. However, the reason why we don't already see them flying around in our neighborhoods as much is because these vehicles are known to create a lot of noise and nuisance. And there are a lot of researchers and companies looking towards mitigating this very issue. But if we need to know the right way to reduce noise, we need to understand the source of this noise. So now I'd like to draw your attention towards the central image, which shows the flow rendering around a quadcopter. You see a lot of complex flow structures or vortices around it. To make it simpler, let's just focus on one rotor in the whole vehicle. Now, this rotor is known to emit noise due to its shape, the upward thrust force it generates, and the way it interacts with the surrounding air. And since all of the flow around it in the real world is turbulent, the problem becomes very complex. And that's where I come in. My main goal is to model this turbulence as accurately as possible, that is, with a high fidelity, so that we understand what is going on at the blade and figure out how that affects the noise generated by the rotor. My turbulence model, when compared to existing state-of-the-art methods, is known to accurately capture the complex flow physics and the highly three-dimensional flow commonly observed in the UAVs. An example of this complex flow phenomena is the laminar to turbulent transition, which is known to create bubbles or laminar separation bubbles, as you see in this picture. They're known to alter the pressure distribution over the blade, in turn affecting the aerodynamic and aeroacoustic performance. So in summary, my research is being carried out to improve our understanding of what exact flow interaction impacts what frequency range of noise, thereby informing the design process. To conclude my talk, I have included some pretty renderings generated by my computational model that shows the flow produced by a single rotor. To give you some context of the computational load, it took about 100,000 CPU hours to generate this image for each rotor. So I'd like to thank VT-ARC for providing me with resources to carry out my research. And I expect to graduate by the end of this year and hope to get into the booming urban air mobility industry that I just spoke about, where I get to build futuristic technology like flying taxis. Thank you.
Thank you. Excellent, Nina. Appreciate it. Lisa, if you could start bringing up your slide while we have time for one question for Nina. I need another brave soul like Eric to ask a question.
Shoot one your way. I was curious when you talk about these rotors a lot, and I'm assuming that because this is going to be some type of an air taxi that needs to have different transitional patterns and things like that, it's going to run into things like birds or bugs or things that are going to kind of like funk up that that's going to kind of funk up that sensor within it. I was curious what you've had to do to kind of like just overreach that or overarch it, or if that's been even a problem that you've had to deal with yet.
Can you repeat the last part? Funk up the sensor? Is that what you meant?
Yeah, or like just kind of mess up what it's looking at in the environment. If it's looking at like this blue sky and then suddenly this bug comes into it, I'm assuming that.
No, you're right. You're right.
Yeah.
So there is a lot of research that's going on where there's bird attacks and stuff. But the thing is, these aircraft are known to fly at lower altitudes. So hopefully that should not be too much of a problem. But the environmental impact is also something that people are bringing up. And that is definitely an active area of research and how that would affect the rotors. But if you see the vehicles, the way they're designed, like in case there is like a rotor failure, there's one rotor that's been that fails because of a bird attack. They're designed to have multiple redundancies. So if one rotor fails, there's like 6 other rotors that can take over and still let the aircraft fly. Yeah.
Thank you very much.
Sure.
Thank you.
Welcome.
Lisa, go ahead.
Hi, everyone. I'm Liza. I'm a 4th year or senior biomedical engineering student, an undergraduate at the University of Virginia, and my research is in kind of co-housed in biomedical and chemical engineering here. And so to start off, my first question for you is, have you all ever played with building blocks or Legos? Just as building blocks can be built into towers, we can build things with molecular building blocks as well. And these molecular building blocks are called amino acids. So my research focuses on how can we form specific tailored peptides from amino acids on an atomic level. And my lab mostly focuses on hydrogels, which are a type of material that you can put cells or drugs into and then input into a patient to promote healing after a wound or injury or illness. And these hydrogels are made up of peptides, which are made up from amino acids, like our building blocks make towers. And based on how we put these building blocks together, they form different structures. So if you have all of the amino acids are the same orientation, they form molecular fibers, as you see on the left. If they are different orientations, they form plates, which you can see on the right. And these 2 types of orientations are called L form and D form. So based on how we put our amino acids together, they can form different structures which have hydrogels of differing properties. And so my work focuses on how we can tune these properties and how these hydrogels come together. So I look at this using complex molecular dynamic simulations. So from an original peptide, sorry, I don't know why this image is not coming up. I'm just gonna exit out. Oh, okay. I'm so sorry, everyone. For some reason this is not. I don't know why I did that.
Unfortunately, I think you just better go on. I don't know.
Yeah. Okay. Well, So basically, we build a, I'm gonna try to, okay. All right. We use the single peptide from our amino acid building blocks, and we can put them into a software called NAMD on UVA's high performance computing cluster. And we put them into a system of peptides in that kind of represent a hydrogel on the computer. And then we can understand the molecular interactions by quantifying hydrogen bonding and pi-pi stacking, which are two forms of molecular interactions that are important to how our hydrogels form. And once we have this information, we can see what the different amino acids orientations impact the hydrogen bonding and pi-pi stacking, and thus the hydrogel components, which we can then tune to treat different applications of different diseases or different cell or drug impacts. Yeah, so we kind of use these computational methods to inform the design of these hydrogels and hopefully be able to tune these hydrogels for different applications. Yeah, are there any other questions? Sorry that this just didn't.
Yeah, we're sorry your presentation didn't work, but any questions for Liza? So how close of this research into actually being put into production, into being actually applied outside the laboratory?
Yeah, so we are currently working on the our lab is starting to put them into mice models right now, but we haven't really brought them into clinical trials yet. So I'd say they're pretty far off, as these simulations are kind of just showing our preliminary understanding of how the hydrogels come together.
Great.
Yeah.
So we'll keep an eye out for it then.
Yeah, thanks.
Let's see. I think Claire is next. I should have mentioned, Claire, you could go ahead and get your slides up.
Yeah, awesome. Thank you. Maybe Wi-Fi at UVA is just not having it today, but I actually don't think I will be able to screen share any presentations.
Well, that's.
Do we have the slides?
Heather, can we put up the slides?
Yep, I'm on it.
Yeah, so sorry about that, guys. I do not know what's going on. Zoom keeps crashing. But a little intro, I guess. Awesome. Yeah. So my name is Claire Cocker. I'm a third-year chemical engineer undergraduate at the University of Virginia, and I actually do work with Liza. So you'll kind of get a different perspective on a different project that we work on. But I just kind of wanted to give you a little background about how we can engineer better tissue or better materials for tissue engineering, and kind of how we're doing that computationally. So with tissue engineering, I'm just going to kind of go from left to right and then down the line. So can follow along that way. So tissue engineering's main goal is to provide healthy cells to an area of damaged tissue. And so one way that we can use this is by using a material called a hydrogel, which is essentially like a jello-like material that you can encapsulate cells into, and then inject by some form of delivery, like maybe injection. You can inject those hydrogels into the damaged tissue area and hopefully have those cells grow into a functionalized tissue. So if we look a little bit closer at what these hydrogels are made out of, they're actually made up of little chains of either polymers or peptides, but our lab is really interested in looking at a peptide called chiral. So it's 5 amino acids long, so it's a very short amino acid sequence. And one of the cool things about chiral is it's self-assembling, so it will go from a bunch of individual peptides and form into a hydrogel. And then we can also tune the properties of the resulting material by using stereochemistry. And so stereochemistry, for maybe non-chemistry people, is essentially the orientation of side chains off of a backbone, so whether everything's oriented to the left or to the right. And so if we're doing this in a, if we have all of our peptides oriented to the left or all of them oriented to the right, we get these really cool fibers that form that make a material that's about the stiffness of a brain that we would like for tissue engineering applications. So if we're introducing stereochemistry, though, where we have peptides oriented both to the left and to the right, we actually get these plate-like structures that form a much weaker material that is not as good for cell growth. So if you want to go to the next slide. Thank you so much. So the way that we investigate this is by using molecular dynamic simulation. So we have a system where we have all of our atoms represented and mathematically mathematically simulating the step and trajectory of each atom in the system. And so we get to see some of these molecular interactions that form in the first few seconds of assembly. And so if we want to continue clicking, if you want to click all the way through, perfect, thank you. So the first analysis that we do is we look at different clustering, which is essentially how well the peptides are coming together and forming kind of like structures together. And we see that over time, if we invert one amino acid at a time, we actually see that all the inversion of one amino acid stereochemistry does not affect overall assembly of these peptides. And so we also wanted to kind of look and try to get an understanding of the intermolecular interactions driving the assembly. And so we see that interactions like pi-pi stacking and hydrogen bonding, which kind of work as sticking factors to stick together these peptides. We see that there are some individual trends that are unique to a specific inversion of an amino acid. But we see that that kind of depends on what the sequence is. And so this is something we hope to investigate a little bit more and really deeply understand. So eventually we can make tunable properties that will allow us to have more versatile materials for tissue engineering applications. So thank you so much for your time. Also, so I'm a third-year undergraduate, so I am starting to look at grad schools, which is I'm really excited about. So if anyone is interested, I'd love to chat about that as well. Yeah, thank you so much.
Thank you, Claire. Any questions for Claire? Marion, if you could be, yep, got you, slides good. Any questions for Claire? So this is an area that I don't have any experience at all in. These chi-fills, are they a naturally occurring thing in the body, or is this a molecule that's been synthesized to try to accomplish a specific purpose?
Right, yeah, so amino acids are safe in the body in the sense that they are natural in that way, but they're synthesized outside of the body. So we use a peptide synthesizer in lab to create those sequences.
Great, thank you.
Thank you so much.
Okay, Marion, take it away.
Okay, so I'm an undergraduate student at Virginia Tech in biochemistry, and I work primarily with positive sense RNA viruses, including SARS-CoV-2 and the dengue virus. And the reason that we looked at RNA viruses is because they have a high rate of recombination, and so we need more efficient drug discovery methods, and we also need broadly effective antivirals. And so I aim to use dynamics-generated structures for drug design because when we look at the dengue virus, we don't have a lot of structural data for it. So on the blue side, we can see the serotype 2 of the dengue virus, and the cofactor in pink is not in the binding site. And we want it to be in the binding site because that's the active conformation of the protein. So in serotype 3, we do have structural data of the cofactor being in the binding site, but we need that structural data for serotype 2. So we perform MD simulations using high performance computing. And the reason that we need high performance computing is because there are so many atoms in this system. In my system, it's about 63,000 atoms that need to be simulated. And so we use HPC to do this. It takes about 150 nanoseconds per day. So it takes 6 days to get 1 microsecond worth of data. And when we looked at a bunch of different analysis, we first looked at RMSD. And RMSD basically just shows the dynamics of the protein over time. And there was a lot of dynamics at around the 600 nanosecond mark. So then we did cluster analysis on that, and we looked at the different clusters of the protein. And there were a lot of clusters. There were 56 clusters in the 550 to 700 nanosecond range. And this is because the cofactor is being extremely dynamic in this part of the simulation. And when we look at RMSF, this shows which amino acids were higher in their dynamics. And we can see that the cofactor amino acids are moving around a lot more and being a lot more dynamic. And in addition, there's another spike that kind of shows more dynamics in the binding site as well. But unfortunately, we weren't able to get the cofactor to swing into the binding site. So we have to perform MD with a small molecule to hopefully get the protein to be turned on and move into that active conformation. Next, we will perform virtual screening on these structures and then test those drugs that we get from virtual screening in a wet lab. Any questions? Oh, also, I'm an undergraduate and I'm going to be applying to PhD programs in the fall.
Great, great. Question for Marion while he's getting her slides up.
I think that Eric has a hand up.
Oh, thank you so much.
And I was muted, so I will start talking again. So I was just curious, it's a bit of a technical question, but it's been a while since I've done these sorts of things. But so what do you do? You know, I see that you use Charm 36 force fields. How much work, and maybe this is work that predates your work in this, but how much work is done to see if that gives you reasonable simulations? And I mean, is there anything done to test for your particular problem? These force fields are giving you good, reasonable results. I'm just curious how that happens.
Yeah, so in my lab, we have like specific force fields that we're kind of told to use because they've been tested and they're good, and our PI knows that they work on the systems that we have. But typically people will choose their force fields based on their system. So yeah, that's kind of how you choose force fields.
Okay. You're already starting to ask graduate level questions, Eric.
Oh, that was purely curiosity. Thank you.
Thank you, Marion. Nee, would you like to start presenting your slides?
Can you see my slide?
Yes, I can see them.
Oh, can I start now?
Yes.
Yes.
Okay. My name is Nhi. I'm currently a graduate student at ODU. My current advisor is Dr. Chen Chung Hao. I'm deeply grateful to Dr. Hongli Wu for his guidance on my initial research while he was working with ODU. Also, thank you, Dr. Danny Uchita, for mentoring this lightning talk. In today's world, with the increase of the cybersecurity risk, it's extremely important to protect online data. Attacker can view, edit, and monitor the transmission, storage, and process the information that is sent to someone online. We can keep our data secure by adhering to confidentiality, integrity, and availability. Lastly, we can have new technology, innovation, policy, and practice in place for end user that they can follow to secure the network and prevent the attack. On my research, I am researching on how to protect electronic data and keep it secure. My research can bring many technology innovation solutions to help our regional, national, and international prosperity of humanity, like education, medicine, defense, security, and many more. Electronic data can be either in the form of image, video, audio, or text. My research is based on the use of reverse steganography method on an image with deep neural network. Using the method, I can decode or reverse an embedded image, video, text, and audio, and read a hidden message that is sent by the attacker. Like you see in this video, there are hiding message behind each video. My research can help find these hidden message. Similar, if I'm sending a message, I can encode it using steganography method and send it in a secure format. I can also divide the image into a graph, which can make it harder to send it in this for the attacker to attack. I'm using the Google Colab Python 3.6 and PyTorch for my programming. I have installed and cloned the deep robot for this project. I have imported all the required libraries for different purposes like NumPy, Cython, MATLAB, and so on. There are many. These are my work solution. So please feel free to contact me if you would like more detail about my research or having any opening for the postdoc after I graduate my doctoral program. So thank you for your time, and if you have any question, please let me know.
Thank you. Questions for me, please. Let's see, Sarah, if you could be putting up your slides. So, Nhi, how how much data can actually be hidden in these kind of images or things? And are you actually trying to find that hidden data, or are you trying to hide the data? I wasn't quite sure.
You can hide the data. You can hide, like for example, on my second slide, you can hide the video, you can hide the text, you can hide multiple images like under the video. So basically, the more you go deep with it, it will make it harder for the attacker to attack into the image. And also, if you convert the image into the binary image, then it will make it easier for the user to defend the attacker.
Thank you. And what's next for you?
After I finished my PhD program, then I would like to go for the postdoc, because I like to have like education job instead of industry job.
Like to go into academia. Okay.
Yes.
Great. Thank you. Sarah, take it away.
Hello, everyone. I am so very excited to be presenting today. Thank you so much for giving me this time to share all of my research, which is probably one of my favorite things to talk about, if I'm being completely honest with you guys. So my name is Sarah Patterson. I am an undergraduate at the College of William and Mary, and I am a computational and applied mathematics and stats major on a mathematical biology track or concentration, as well as a biophysical chemistry major. So during this talk, we're really going to be talking about how I'm severely putting that computational part of my degree to work. So this is actually going to be my first time down here visiting William and Mary. And a large part of the reason why I'm here is just this computational major. I was so intrigued. I've never seen something like it. And it's absolutely what I want to do from here on out. And then this is my absolutely lovely and terribly brilliant PI, Dr. Margaret Saha. So a lot of what I do in the Saha lab is going to be genetic analysis and what genes an organism turns on and why do they even want to do this. So much of my research is looking at this frog, which is not a terribly pretty thing, but luckily I only have to look at it on a computer. But what's so intriguing about this frog is that the way that it undergoes its neurodevelopment is extremely similar to ours, which is fascinating because we look vastly different and we live in terribly different environments. So it's so intriguing for us to better understand how is it turning on these genes, where are they going, and there's so many different things actually happening inside every single cell at a time. So a lot of my job is figuring out what is it saying and how is it saying it and why does it want to say these things. So the way that part of what I am doing is I am going through what's called a signal transduction pathway. So if you've taken an intro bio class, I'm sure that this has just been drilled into your head, but it's this idea that a cell has to communicate with all of the cells around it. And most of the time it does that with genes, but these genes are going to turn into proteins or different chemicals, hormones. They're going to go and talk to all of their friends. And this communication is a huge part of why that frog can be an embryo into this entire full-size, silly looking thing at the corner of my screen. So a lot of my research is pertaining on to actually taking these individual genes as they are getting expressed inside of every single cell and then quantifying them and figuring out how much is it expressing X gene, what is X gene trying to communicate to the rest of the organism, and how is this really developing this entire brain. So as we can see over here, we have these what's called RNA sequencing reads, which are just different transcripts that we're looking at that correspond to genes. And then through a whole heck of a lot of work, we're taking all of these individual genes, we're finding where they belong in the genome, how often are they expressed, how often are they not expressed, and then we're going to put them into a map that clusters it, and it looks extremely similar to the one down here, where we're actually characterizing all of the cells and figuring out which cell is a whole heck of a lot like one, or which one is a whole not like that one, and where do they belong and what's their job inside of all of these things. So I use various different types of softwares to help me pre-process this data into different coding environments. Is a lot of what I like to do.
And what's next for you?
I'm hoping to go to grad school. So I'm still an undergraduate. I am just a sophomore too. But go to grad school with whoever wants me.
Great, great. Let's see, one question for Sarah while Cynthia's getting her slides up.
Question. So in your UMAP there, do you do time resolved single cell RNA-seq? Like, are you following the development of the frog throughout its embryonic process? Or...
Yeah, that's a fantastic question. So to try to make this into a thing that a lot of people who maybe don't have a biology background can better understand, we're doing a couple of different experimental types with the single cell RNA sequencing. One of the things that makes it really hard is that we are multicellular organisms through in and throughout. All of our cells really want to talk to each other, but in order to understand how they're talking to one another, we actually have to take them apart single cell by single cell, which makes one of the things very difficult because they're like, oh my God, if I can't talk to my neighbor, I think I'm just going to die. So part of our thing is first off, trying to make sure that we can get good data and we're not ruining the transcripts or the genes they're trying to express. And then part two is just to prove that we can do it. My current data is working with an embryo that's really really early in its development, and then comparing that to one that's a little bit further along the process of it, but not yet fully grown and independent outside of the egg. And then the real nitty-gritty experiment that we're looking to do is truthfully to flip its nervous system, which is that brain that's really similar to ours, and seeing that if we can flip it like totally, like just on its axis, it doesn't respond, like it pretends that you didn't even do it, it develops totally fine. So that's another really interesting thing that we're looking at doing. But absolutely, we're taking them some different stages and then comparing different things that these frogs can do that we can't, to just the way that they grow up and live.
Thank you. Thank you. Cynthia, please.
All right. Good afternoon, everybody. I'm Cynthia Cias. I'm a third-year PhD student at Virginia Tech, and today we're going to be talking about cover crops. Cover crops are typically a crop that follows a cash crop, such as corn or soybean, and they have multiple benefits. And so the use of cover crops has steadily been increasing in response to herbicide resistance issues. And so there is a lot of previous research that shows that cover crops have the potential to suppress even some of the most troublesome weeds, such as Palmer amaranth. And although we know of the benefits of cover crops, such as weed suppression, there are still a lot of questions that remain and that we get from our growers. And so the objective of my research is to answer three main questions that we receive from Virginia growers. Question number one is, how much cover crop biomass do I actually need in order to suppress weeds? In order to answer that question, we established a low, medium, and high cover crop biomass gradient that was established by staggering planting dates in the fall. Question number two that we get a lot is, should I roll my cover crop or should I leave it standing? In order to answer that question, we had a rolled and standing cover crop treatment, as you can see there in the middle. And then finally, when should I terminate my cover crop? To answer this question, we had both a brown and green cover crop termination timing. A brown timing is when you terminate cover crop 2 weeks prior to planting, and a green termination is when you terminate the cover crop at the time of cash crop planting. And so what we found from this factorial experiment was that out of the three factors that we evaluated, the main effect of cover crop biomass proved to be significant in reducing Palmer amaranth emergence. So as you can see on the graph at the top right, we see that negative relationship that shows us that as we increase our total cover crop biomass, we also see a reduction in Palmer amaranth emergence. And these results are similar to other results in literature that we've seen in the past. And so that's really good information to share with our growers. Although we were not able to make strong conclusions as far as the rolled or standing or the termination timing aspects, we were able to shed some light as to some of the complexities of cover cropping. We found that when we were planting the cash crop, in this case the soybean crop, into the cover crop biomass, we had trouble making seed-to-soil contact with the planter, particularly under high cover crop treatments. And so that kind of information is also very important to share with our farmers who are interested in cover cropping. The practical implications of this research involve sharing with our growers that although cover crops are a strong tool in integrated weed management, they need to be managed properly in order to maintain crop yields that are profitable and sustainable. And so this is all very applied work that is easily translatable to our growers. Thank you for listening, and I'm happy to answer any questions. As far as future plans, I'll be graduating in December, and I hope to find a job with industry and agriculture.
Thank you.
Two hands raised.
Question for Cynthia while Afrina is putting up her slides.
Yes, I have a question, Mark. Cynthia, the question I have is, I'm just curious. So the cover crops after the harvest, are they being incorporating back to the soil, or they are being clear out?
So typically, you'll terminate a cover crop either chemically with an herbicide or mechanically by rolling it, as I showed in that second set of pictures. In terms of incorporation, a lot of our farmers, especially around Virginia, are no-till farmers, which means that they are not tilling the ground. They're pretty much just planting into it. And that's why with the heavier cover crop biomass, a lot of times that planter isn't putting enough force. And so that's why it's important to communicate this to our farmers. Did that answer your question?
Yes, thank you, Cynthia.
Thank you, Cynthia. Afrina, your turn.
Afrina, I think that you are in the presenter mode.
Exactly.
You might want to switch. Yeah, there. Perfect.
Okay, go ahead, Afrina.
Mute. Oh, sorry, I was muted. Okay. So hi, I'm Afrina Tabassum. My PhD thesis is on a multimodal learning, representation and generation. More specifically, I will be talking about one of my projects that will be improving self-supervised learning for multimodal data. And the main motivation for this work is building improved model for contrastive self-supervised learning for multimodal data. And by multimodal data, we are working with audio, text, image, and videos. Why we choose these four modalities are because these are the four most common modalities in multimedia communication. And using these modalities, so we train different intelligent assistants, for example, Cortana, Amazon Echo, and all the other virtual assistants. So what we do is that we use this data to train these models to learn to be like acting like a human. But and the most forward way to do this is using supervised learning, that we have some labels, we have some data, and we train the model to learn the mapping from data to the label. But the problem is supervised training requires labels, and which requires a lot of time and also manual labor. So there comes the self-supervised learning. Self-supervised learning, most importantly, contrastive learning, does not require any labels to train the model. For example, we have an anchor, that is one data examples. And we have a augmented example of that anchor. That means like a cropped or like grayscaled, different or using different augmentation technique. We call it a positive sample. And all the other examples in that data set will be considered as negative examples. So what contrastive learning does is minimizes distance between anchor and positive and maximizes distance between the anchor and the negatives. So the question comes can we use all the other examples as negatives? The answer is no, because we have millions of data, and we cannot use all the examples as negatives. So in our model, in our paper, we select optimal negative examples based on three properties. First one is similarity with the anchor, and second one is influence of the negative examples over the anchor, and third one is diversity of those negative examples that has been selected. And we have performed approximately 2%. We have achieved approximately 2% improvement in 3 benchmark image classification data sets, and 10% improvement in 5 out of 6 text representation tasks. So yeah, that is the end of my presentation. And I want to thank ARCA to support the experiments of my papers. And after finishing my PhD, I would like to explore the industry research and go and do some, hoping to do some amazing things, I guess.
Thank you, Afrina. Rashid, if you would put up your slides. We have time for a question for Afrina.
I have a question.
Yes, please.
I was going to ask, how do you measure the distance between images or from the categories? I'm not sure if you're measuring on the image side or on the categories.
We are measuring the distance in the feature space. That is, we like map the image into a like smaller dimension features that will be like 100 or 500, and then we measure the like distance. Distance can be measured with similarity and cosine similarity or Euclidean distance. Like it depends on like we can use any one of them.
Great, thank you.
Thank you. Okay, Mashid. You may start, Mashid.
Sorry, I was muted. I lost my shark. Have you seen it? My name is Mashid Ahmadian. I'm a PhD student in statistics at the Virginia Commonwealth University. And today I'm going to talk about my research. So I'm working with a big data set, which is collected by a group of ecologists. They attach some tags into some salmon sharks' bodies and track them using satellites through the Pacific Ocean. So, for days that the salmon sharks swim on the surface of the water, the satellites were able to track the location of the sharks and record the coordinates. But for the days that the sharks were unobserved on the surface of the water, we do not have any information of where they were swimming. So here, let's look at this map. Here it is Alaska, and this is Pacific Ocean. And these little red dots are the coordinates of just one shark over 6 months. So more specifically, let's look at this circle. These dots are the coordinates for the shark, the coordinates of the location of the shark for almost every day, except for 3 days here that the shark was unobserved. So I developed an algorithm. This algorithm is interpolating the approximate location of the shark for the days that it was not observed. So let's look at this heat map here. This heat map is a simple example of how this algorithm actually works. These ellipses here are showing the approximate location that algorithm is producing, with considering that this red color is showing the most probable location for the shark. So with over 100 salmon sharks, and the look that's been tracked for more than a year, and for almost more than 10 locations recorded every day, we have a very large data set. And this algorithm is computationally intensive. So a personal computer cannot perform it efficiently. And we need to use high performance computing for that. As I mentioned, I'm a second-year PhD student at VCU, and I'm expecting to graduate in 2 years. And I would like to work in fisheries management after I graduate. Thank you for your attention.
Thank you, Mashid. Very interesting. Do we have any questions for Mashid?
I have a question.
Yes.
What is the sampling frequency for the red dots?
What do you mean by sampling frequency?
How often do you get data from each individual shark?
So as I said, they're recorded almost every day, and like so for days that the salmon sharks swim on the surface of water, It records like 10 to 20 locations. It depends on how many times the satellite could track them. But there are like more than even 10 days sometimes that the shark was not seen over the whole ocean.
Okay. So if they were on the surface of the water for all 24 hours, I know that's not true, but if they were on the surface of the water the whole time, would you get about 20 data points?
I'm not sure about that because
Okay.
I didn't collect the data. I'm just using it to develop the algorithm.
Okay. Sorry.
Thank you. I really want to thank all of our presenters. This was very, very interesting to me. We do have a few minutes, so if somebody has thought of a question for one of our presenters that they didn't get a chance to ask, we do have a couple of minutes, so we can probably take 2 questions in the time that we have remaining.
If no, Mark, I think that we would like to, Eric would like to announce that we have a winner. We have the winner for the Woman in HPC, Virginia Woman HPC logo.
Okay, turn it over to Eric.
Hi, everyone. So the winner was Miguel Montalvo. Unfortunately, at the last minute, he actually had another engagement he couldn't get out of, and so he's unfortunately not here. But so he was the winner. The logo is shown on the screen here in the bottom right. And we will be trying to incorporate it where appropriate into our website and other uses in the future. And so, congratulations to him.
Thank you.
All right.
Just by a rough show of hands, how many of you, is this your first time at a Virginia Women in HPC meeting? Just raise your hand on the screen so I can kind of get a rough idea. Okay, still coming in. Looks like we've got 6. That's about 1/3 of the people that are attending. Oh, a little more, 1/5 now. Okay. Well, we really appreciate you joining us today. We have these meetings every month, and whether you are a woman in HPC or someone who supports a woman in HPC, we would encourage you to continue to remain involved, to go to our website and keep an eye out for events coming up, or to join the mailing list, and we will keep you informed of the different activities. Thank you all to our presenters. Appreciate the time that you put into. I would like to also comment the fact that you were very punctual. I didn't have to really give anybody the red light. You did a fine job. So, thank you.
Thank you. Let's thank the people that help with this event. We have Haley, Heather, and Rupa. And I just want to say really thank you to all of you for helping to put this event together. If this is something that would you like to see in the future, please leave your comments or Google Doc. If not, you can do it in the survey that we're going to send out with the recordings and the presentations of the students as a follow-up. But thank you everyone for being with us, and I hope you had a great time.
See somebody's hand up.
Mark, I think that you can stop the recording.
Okay.